Good day, sir. My name is Alrose Rivera from the Group 1, which talks about the unicellular eukaryotic microorganism. And I am going to talk about the third and final supergroup of the eukaryote, which is the Excavata. Now, what is Excavata? Excavata is a significant group within the eukaryota domain, characterized by a diverse organism with specific traits like a feeding group. They exhibit various lifestyle, including the free-living and the parasitic forms. There are three key characteristics of Excavata. The first one is the feeding group, which have a distinct feeding robe where they might ingest food particles. This groove, which is usually found on one side of the cell and is connected to the specific feeding-related components. The second key characteristics of the Excavata is the flagella. In euglenozoans, these flagella may have a crystalline rod that makes them stand out. With the help of their flagella, many excavates can move around. The third key characteristics of Excavata is the diverse lifestyle. The free-living, parasitic, and symbiotic ones are demonstrated by the excavates. Their roles and adaptations in the environment reflect this diversity. Now let's move on and talk about the four well-known subgroups of Excavata with significant ecological and human health importance. The Fornicata, the Parabasilids, the Euglenozoans, and the Diplomonads. Now let us first discuss the Fornicata. Fornicata, which is characterized by lacking typical mitochondria and possessing hydrogenosomes, but instead, they often have multiple flagella and two equal-sized nuclei. Naegleria fowleri is one of the prominent examples of Fornicata that is well known for its pathogenicity in causing infrequent but serious brain infections. Fornicata's notable features and adaptations is a plant that relies on hydrogenosomes for energy production and can survive in anaerobic environments due to their adaptability and ability to survive in low oxygen concentrations. The second subgroup of Excavata is the Parabasilid. Parabasilids is characterized by the presence of the parabasal bodies, flagella, and the lack of conventional mitochondria. They often exhibit unique morphology, such as an undulating membrane. Trichomonas vaginalis is the common parabasilid parasites, which cause of trichomoniasis, an infection that is contracted during the intercourse in humans. Parabasilid's ecological significance and impact, including Trichomonas vaginalis, are essential for ecosystems, but their negative impact on human health underlines their ecological value. Now, the third subgroup of Excavata is the Euglenozoans. Euglenozoans is characterized by the distinctive feeding groove, two flagella emerging from a pocket to one end of the cell, and the presence of the chloroplasts in some members. They also possess a unique crystalline rod inside their flagella. Euglena is one well-known example of Euglenozoans, and it is typically found in a freshwater habitats. Euglenozoans' distinctive features is a crystalline rods that facilitate movements and stability, while their chloroplasts enable certain species to perform photosynthesis, enhancing their ecological adaptability. Moving on to the fourth and last subgroup of Excavata, the Diplomonads. Diplomonads is characterized by having two nuclei, multiple flagellae, and modified mitochondria, called mitosomes, instead of the typical mitochondria. Now, Giardia intestinalis, or usually referred to as a Giardia lamblia, the widespread of diplomonad parasites, causes the diarrheal disease giardiasis in humans. Diapogonads' significance and ecological functions, like Giardia intestinalis, pathogens causing gastrointestinal illnesses, significantly impact human health and promote microbial diversity and nutrient cycling in various environmental settings. And I guess that ends my part. Once again, this is Elrose P. Rivera from the Group 1. That's all. Thank you.